Welcome to the Proteomics course. Today we will talk about matrix assisted laser desorption ionization, time of flight, MALDI TOF. In previous lecture, we talked about basics of mass spectrometry, the various combinations of mass analyzers and ionization sources. Now, it is time for us to combine those and start discussing these in more detail. So, today let us focus on the MALDI TOF, which is one of the very widely used technique in proteomics. This provides a high throughput platform for several applications, including molecular weight determination, protein identification, as well as post translational modification studies. Let us first start about basics of MALDI TOF. So, MALDI is an efficient process for generating gas phase ions of peptides and proteins for mass spectrometric detection. MALDI is one of the most widely used ionization technique currently applicable in the proteomics area. This ionization method was independently developed by two scientists, Koichi Tanaka and Hillenkamp. Tanaka also received the Nobel Prize for his novel contribution into soft ionization technique such as MALDI. So, let us go through some of the basic concepts involved in the MALDI TOF. We can split that in two parts. One is MALDI, which is ionization source, and other is TOF, which is a mass analyzer. Let us first talk about matrix assisted laser desorption ionization or MALDI. So, analyte or the proteins of interest are mixed with the matrix, which is usually an aromatic compound. There are various types of matrices available, which we will talk in more detail when we come to the sample preparation and matrix selection. But just for your reference, we can use 2,5-dihydroxybenzoic acid, we can use cinepenic acid, and there are several other choices. Once you selected a matrix for the experiment, then analyte and matrix can be dissolved in an organic solvent, after which then it can be placed on the metallic target. As you can see in the slide, the first left section shows you how to place the analyte and matrix together on the sample plate. Now, once you have placed the matrix and the analyte on the target plate, you can put that in the vacuum chamber and apply high voltage. Now, these crystals are targeted with the short laser beams, as you can see in the slide. Then rapid sublimation can convert analyte into the gas phase ions. Now, these ions once generated, they can accelerate away from the target plate through the mass analyzer, which is time of flight TOF tube, and they can reach towards the detector. Sample preparation and spotting. The protein sample must be prepared suitably before it can be analyzed by the mass spectrometer. If you have run a 2D gel, so first of all, the purified protein of interest need to be excised from the gel on which it has been electrophoresed and dissolved in the suitable buffer. So, depending upon the application, if you have purified a protein, you can separate that on the gel and then cut that band, or if you have a mixture of the proteins in 2D gel, you can just excise that particular spot. A spot can be dissolved in the suitable buffer. Trypsin is then added to this mixture in order to carry out digestion of the protein. Trypsin cleaves the protein at the C terminal of the arginine and lysine residues, but that is not always universal. If you have a proline present immediately after, then it will hinder that. But overall, the protein is digested into smaller fragments of manageable size. Once the protein sample has been digested, all the salt, buffer and any detergent must be removed from the sample. So, after doing in-gel digestion and before proceeding for the mass spectrometry analysis, in between an efficient step is to use some filters or zip tip which can eliminate some of these contaminants and salts. It offers several advantages such as quick purification, sample enrichment and ensures that there is no contamination. So, there are multiple advantages of using zip tips. However, it can purify only limited volume of the sample and also it adsorbs some amount of the protein sample, thereby leading to losses. The purified protein sample can be mixed with an aromatic matrix compound such as alpha cyano 4 hydroxy cinnamonic acid or cinnapinic acid in the presence of an organic solvent. The components are mixed thoroughly and then the solution containing the organic matrix with the embedded analyte of interest can be spotted onto a metallic MALDI sample plate. MALDI gives you an opportunity to analyze large number of samples in high throughput fashion. The target plate containing the spotted matrix and analyte can be further placed in a vacuum chamber with high voltage and short laser pulse are applied. The laser energy gets absorbed by the matrix and is transferred to the analyte molecules, which undergo rapid sublimation resulting in gas phase ions. The gas phase ions generated are accelerated and travel through the flight tube at different rates. The lighter ion moves rapidly and reaches the detector first, while the heavier ions migrate slowly. These ions are resolved and detected on the basis of their mass to charge ratio and a mass spectrum is generated. Parameters such as geometric design, power supply quality, calibration method, sample morphology, ion beam velocity etc. all of these factors affect the accuracy of mass detection. So, now you know how to perform the cleaning step by using zip tips. Now, you have the sample ready and you have selected the matrix. So, now let me show you this various steps involved before you can actually start the MALDI experiment. So, you need to select the matrix, you need to prepare matrix, you have already done the sample purification. Now, sample need to be deposited on the MALDI plate. Either you can mix with the matrix or you can do these two separately. There are various combinations one can try, and then once both sample and matrix is deposited on the MALDI target plate, then you are ready to do the drying, and then plate can be used for MALDI TOF instrument for further analysis. Let us first talk about matrix selection. So, the important step in MALDI TOF analysis is selection of appropriate matrix for the sample. The matrix selection mostly depends on the molecular weight of the target to be analyzed, and often the type of application which you intend to do by using these instruments. So, these matrices are low molecular weight organic compound with low vapor pressure and volatile nature. Most of the matrices are acidic in nature, so it can easily excite the photon and ionize analyte for the analysis. However, there are few basic matrices are also available. In the slides, I am giving you an overview of few matrices and some of their properties, but there are many more properties which is not mentioned here, but just to give you certain major features of these matrices commonly used for the various applications. So, one is the alpha cyano 4 hydroxy cinnamonic acid. When you have peptides less than 5000 Daltons or lipids and nucleic acid, one can use this matrix. One can also use cinnapinic acid if peptides and proteins are having more than 5000 Daltons, and it can also be sometimes used for the lipids. Then you have options such as 2,5 dihydroxybenzoic acid, also known as DHB, small molecules and peptides which are not ionized by the other molecules can be analyzed by using this matrix. Trihydroxy acetophenone THAP, this is used for small nucleotides and also used for phosphorylation and specialized applications. Then we have picolinic acid, which is generally used for the nucleotides. So, these are only few representative matrices. As you can see, there are many options available for selecting the matrix depending on the molecular weight and the type of applications. But regardless of these matrices absorb energy from the laser source and converts both matrix and analyte into the gaseous phase. Matrix can also ionize analyte molecule by providing energy which comes from the laser bombardment. Now, once you have selected a matrix, matrix can be prepared by mixing it into a suitable solvent and vortex it for few minutes, so that it can dissolve properly. Now, you are ready with both your analyte, the protein which you want to analyze, as well as the matrix which you have selected for your application. Now, one need to think how to deposit that sample on the MALDI target plate. So, there are many ways of deposition of sample and matrix onto the MALDI plate. Mostly sample and matrix are mixed in a Pandolf tube, and then the mixture is directly deposited by using a micropipette onto the MALDI plate. But one can also try various combinations. In one approach, the sample is first deposited to the MALDI plate, followed by the matrix is deposited above it, and then it is properly mixed before drying process can happen. Other way of doing is to apply that with the sandwich based method, in which first a small amount of matrix is deposited on the plate, then you add the protein sample, and again the matrix is spotted on top of it, so that you have enough matrix in the below and the above of the analyte. So, one can try different combination of placing the matrix and the analyte. And then once you have placed all of this sample of interest on the MALDI plate, then you are ready to dry the target plate. Software spotting is done and MALDI plate is dried almost 30 minutes. Then the instrument can be turned on and MS analysis can be performed. Now, there are various type of configuration of these instruments available, as well as there are various type of commercial software which help to operate the hardware. It is not possible to go into individual detail, but I am going to show you the generic steps in the following video of MALDI TOF instrumentation. MALDI is performed in two steps. In first step, the compound for the analysis should be dissolved in a solvent containing small organic molecules known as matrix. This mixture is dried before analysis and liquid solvent used in the preparation of solution is removed. So, in this video, by depicting the matrix preparation as well as instrumentation, we will try to give you overview of the MALDI TOF instrumentation. So, spot the mixture on the MALDI plate. How uniformly you can plate these mixtures on the MALDI plate ensures your good spectra and data quality later on. Completed on the MALDI plates, the samples are allowed to dry for 30 minutes. After which the instrument is switched on and MS analysis can be performed. While these steps are happening, you need to ensure that instrument is on. So, click on the software and open the acquisition window and then click on the open door. Insert MALDI target plate, face up with the cutoff corner to the front, and now by using software, close door. The door of insertion chamber is now closed. You can select the plate, you can view the overall plate on the screen and then select the spot which you want to analyze. So, click on the yellow target in the acquisition window and select go to the location. You can now do the laser bombarding and peptide spectrum is generated. We have shown here one standard protein, bovine serum albumin. So, you have to look at various location where you can get best spectra from that spot and then you can freeze it. Same process can be performed for different spots and different regions. Now, we have shown here a spectra for the PEP mix. So, now you are clear with how to perform the MALDI TOF experiment. Now, let us add one more mass analyzer. So, now we have a configuration of MALDI TOF. So, MALDI can be coupled to the tandem time of flight in combination with another time of flight. So, TOF or with hybrid quadruple time of flight analyzers which are separated by the collision cells. Now, for proteomic application, it is recommended to use the TOF or QTOF. The peptide ions are accelerated through the first time of flight tube, as you can see in the slide, and then they are dissociated by introducing an inert gas into the collision cell. This process allows the collision induced dissociation spectra from the MALDI produced from the precursor ions. Now, these hybrid configurations are more sensitive than the triple quad and the single time of flight. So, the combination of TOF allows the protein identification through the peptide mass fingerprinting, and high throughput analysis of the protein or proteome is possible with the hybrid TOF analyzers.
Open the Matrix Science browser window to carry out online data analysis. For peptide analysis, click on Peptide Mass Fingerprinting. Mascot Peptide Mass Fingerprint. Please enter the user ID and details to acquire information in case of any network loss. The following parameters should be selected. Database. Primary databases include SwissProt and NCBI. Select SwissProt database for data analysis. Enzyme. Select enzyme as trypsin. Missed cleavages. Missed cleavages are allowed up to 1. Next is taxonomy. The protein extracted from the biological specimen has to be assigned to a particular species or a group of species to which the sample belongs. When you are not sure of organism, select all entries. Fixed modification. Select carbamidomethyl. Fixed modifications are applied collectively across the database to account for change in mass of specific residue. Now scroll down and select oxidation. These are mass changes suspected to occur during sample handling and accounted for by increasing the number of primary sequences compared against experimental masses. Include it as variable modification. Depending upon user needs, the parameters can be changed. Protein mass is the mass of intact protein and is optional. Set peptide tolerance as plus minus 1.2 Daltons. Mass values. It specifies the type of charge to be examined. Select MH positive. Select monoisotopic. Report top 10 hits. Select decoy for statistical analysis. Copy the M by Z values and paste in the selected box for mascot search. Click on Search. Data output. The output can be seen in 3 sections. In section 1, the summary of set parameters defined by user. Section 2, mascot score histogram. The number of protein hits with score is plotted along the graph. Section 3, summary report in which matched proteins from the database with the details of important parameters are displayed either in concise format, protein format, and the data can be exported too. Data analysis. If the search parameters are not the best fit, the software generates an error message. Depending on the error message, user needs to change the parameter setting and do the search again. In section 2, mascot score histogram, the number of protein hits and their score is displayed along the graph. Protein view section displays matching of the query peptide to the protein sequence in the database. The sequence type, the matched region, what is the expected and calculated values of the query peptide and sequence details. For better protein identification and to increase protein score, CID of each peak generated is carried out to generate MS-MS data. For such data analysis, MS Iron search option is selected from the Metric Science browser window. In MS-MS search tool, more input parameters like quantitation, MS-MS tolerance, peptide charge, instrument, etc. in addition to fields for PMF and rest other parameters are similar to that of the peptide mass fingerprint. Depending upon the process carried out for data generation, a selection in the quantitation must be made. In case of label-free quantitation, select None in Quantitation tab. Remaining parameters remain same as PMF. Browse the MS-MS raw data file and search the results using mascot. Define the instrument that has been used to generate the raw data. When we don't know the name of the instrument, select default. The result output generated is almost similar to PMF output. The accession ID indicates the protein information obtained from database, marked as green. The protein hit outside the green box indicates p-value less than 0.05, which is statistically significant, whereas hits inside the green box indicates random matches. Mascot search results. Score. The peptide score is a sum of highest iron scores for each sequence with duplicate matches being excluded. Nominal mass. It predicted mass of the protein. Calculated PI value predicted isoelectric point of the protein. Sequence coverage indicates the percent of matching peptides. All the peptides are displayed with matching peptides indicated in red. The protein view obtained on selecting a particular protein link is very similar to protein view observed in PMF. It provides detailed information about each of the matched peptides displayed. The start end and end position of amino acids calculated and experimental molecular weights, number of missed tryptic cleavages, sequence of each peptide fragment and their corresponding iron scores are shown. The highest iron scores are used for computing the final protein score. Each peptide in tandem MS-MS undergoes a second round of fragmentation when it passes through the second mass analyzer before it reaches the detector. This provides significantly larger amount of information regarding each peptide fragment, which can be viewed by clicking on the peptide links provided in the summary report. The fragmentation pattern is displayed graphically, which can be zoomed into as per the requirement.